Where's the chat? Good morning. Can anybody hear me?
Yep, I can hear you.
Okay, good. If you want a record that you're here, put your name in the chat. Okay. We'll wait a couple minutes before we start. It's not quite 9 o'clock, so I'll just kind of stall. Let's see. Oh, I should say something about tomorrow's lab. Not tomorrow, Friday's lab. I have a meeting. I'll be in my office on Zoom in a meeting for much of our lab. But we'll still have lab. I will set up stations, and hopefully I'll have stuff on the board written for you and some handouts. So be there on Friday, and I will be there for short bits of time, but I can't be away from the meeting for too long. So we'll be there in person on Friday. There'll be handouts, color handouts. The only time I ever give color handouts will be this week and next week. So you'll want those to study from, because that's why we have a black and white manual, because we have color handouts. Okay. All right. Let's again start in the chat if you want to be. Put your name in the chat if you want credit. I'm going to share a screen and we'll look at the schedule. Let's see here. Okay. Does anyone see the lecture topics and assigned readings for human anatomy and physiology?
Yes.
Okay, thank you. All right, so we just finished exam 2. The rest of the course is all the nervous system. Oh my God. So chapter 12, 13, 14, 15, and 16 is all nervous system. So we'll do 3 chapters, and then we'll have an exam, and then we'll do 3 more chapters, and then we'll have the final exam. But the nervous system is really complicated, and so these chapters we're all going to go over pretty closely. And so it'll be a week each time, lots of new stuff, and lots of important stuff for the next semester as well. So it's important, and lots of detail. So it'll be that. I should get your last exam kind of corrected by the end of this week, maybe, and then those will be available for you to see. So. Okay, and let's share another screen. Let's look at this week's study guide, because there's a lot of new stuff here. So this is, I think, the biggest chunk of words I've given you. And even some of these are just abbreviations. So PSP is postsynaptic potential, and then it can be excitatory or inhibitory. So there's other things there. Lots of this is the real meat and details is how nerve cells function. And so a lot of this is about how action potentials go in very specific ways. And so it's kind of important to recognize that's where the really hard stuff is, but it's not the only stuff. And so that's only part of this chapter, but it's a big part, and it's the hard part. So let's go look at some of the other parts, and we'll work our way to that other stuff. And so, perhaps it's not as hard, but it's important, is just this organization and general function of the nervous system type stuff. And so let me get to that. So the nervous system, the general function. So just like the skin and the muscles and the bones, I asked you in the exam on those general functions. This is important. It's a little slide and it's not any hard words, but it's an important part. So the nervous system carries sensory input. And so that's a big function, as it carries sensory input from outside the body, sensory input from inside the body. We have lots of sensory input. Some of it we're aware of, much of it we're not. You know, like our stomach is reporting back to part of our brain on our meal, what was in there. We never, you know, we don't know that that's going on, but the brain knows, okay, there was carbohydrates in that meal, so it knows what to secrete into the digestive tract. There's lots and lots of sensory input. Okay, and then the brain also, or the nervous system also has motor output, and your body or your textbook calls this response. I refer to it as motor. And what that means is that it's activating muscles and glands. And these muscles and glands, again, you know, we're aware of the muscles when we activate our skeletal muscles, because those are voluntary, but we have other muscles that aren't voluntary. These open and close arteries. We have muscles in the digestive tract that move food through. We have muscles that are working all the time that we don't know. And likewise, we have glands that are working all the time, and we're not aware of that. Our digestive glands work, our you know, various glands are working, and so this response activity is usually directed by the nervous system. And then there's this big category, integration. Okay, we have lots of sensory inputs coming in. The body has to integrate them, so it has to. Those sensory inputs come in and they're weighed, and then whether or not or what action is determined. This integration is you know, we've talked about this with homeostasis, with temperature control. The hypothalamus is receiving information from lots of different places, skin receptors, you know, receptors on blood temperature, and the skin weighs that or the brain weighs that and decides what to do. And so this is integration, and this is a big part. And integration includes our consciousness, memory, our thoughts, our feelings. That's all integration. Okay, so they just break it down into just 3 little functions: sensory, response, or motor.
Friday, he has some meeting during our lab. So he's like, I won't be in lab, but go because gonna have like handouts and stuff set up and stuff.
Okay, all right. Is that a question, or are you talking to someone else? Oops, okay. Okay, all right.
This
All right.
lady's losing her shit because he said he hasn't graded our test yet.
Hello?
Whoever's talking, put y'all thing on mute.
Excuse me, hold on. Hold on, let me, I have to back out of this and then I can mute them.
Swear to God, she makes me feel like
Let's see here. How do I get out of it? I'm gonna mute people.
I don't know what.
Who's not muted?
It's Kirsten.
Yep, okay, there we go. Sorry about that. Okay, where was I? And I'm there. Okay, so this is just a little bit in the front of chapter 12, but it's important. Likewise, there are other parts of that are pretty important that are small. So they're talking about the parts of the nervous system. And we can divide the nervous system up in anatomical ways. The central nervous system is the brain and spinal cord. It's encased in bone, floating in cerebral spinal fluid. This is where most integration takes place. Okay, and then the peripheral nervous system is nervous tissue outside the central nervous system. It's mostly sensory receptors and nerves. This is most of where that sensory response stuff is going, and there's very little integration in the peripheral nervous system, but there is a tiny bit, and sometimes it's important. So these anatomical divisions perform different functions, but they are anatomical divisions, okay? The peripheral nervous system has divisions as well. The peripheral nervous system has a sensory division and it has a motor division. We can also divide the motor division up into somatic and autonomic. Okay, and when we talk about somatic and autonomic, somatic is that stuff we're consciously aware of. That includes the skeletal muscles. The autonomic includes things that we're not consciously aware of. They're mostly homeostatic mechanisms that are controlling things like cardiac muscle, smooth muscle, glands, and things like that. The autonomic division also includes an enteric nervous system, which controls the digestive system. I'll say a lot about autonomic systems later. There's a whole chapter on it in our textbook. Okay, and so this diagram, this is a nice diagram that kind of shows central and autonomic nervous system, peripheral nervous system, sensory, motor, you know, things like that, somatic and stuff like that. So this is an important kind of thing. We'll refer to these terms and we're referring to parts of things. And so that's important. Likewise, I went through, hold on here. I went through an example here. And this is an example that shows different parts of the nervous system acting at different times. So peripheral nervous system, seeing a stimulus is something that initially happens with sensory receptors and sensory nerves. So it's in the peripheral nervous system. So if you follow the bear down, you get to sensory division. And this conducts action potentials from the peripheral nerves to the central nervous system. And then in the bottom, in the pink, central nervous system integrates this information. And it initiates the response. And so you get integration and response started. And the response here is going to go into motor activity. So it's motor. And then there's two arms of the response. There's a conscious arm where you decide to run away and you activate skeletal muscles. You're consciously aware of all this. And then there's an autonomic part of it that you're not aware. And that's a part that releases adrenaline. It's a part that increases your respiratory rate. It's a part that will activate smooth muscles and glands. You'll start perspiring. And so you have a change not only in your somatic output, but also in your autonomic output. And so this is there. And of course, running away from a bear, we should all know bears are faster than people. That would be a very bad idea. Hopefully your integration center would say, don't run away, back up slowly. Okay. So these parts are important. These are important parts. I will ask about these divisions. I will ask about these functions. It's all covered in a very short section in the beginning. It's definitely understandable, and it's definitely something that we should be able to master pretty easily, these different divisions. It'll help you if you get this right away. Now, the next part is the hard part. The next part isn't more important, it's just harder, okay? And it's really on the cells, the tissue type stuff of the nervous system. Okay, the functional cells are neurons. The neurons have a distinct appearance. They're extremely large and long-lived cells. They have a good-sized cell body, and then they have two types of extensions, dendrites and axons. Dendrites carry information to the cell body. Axons carry information away. And so here they show just one long axon. You can have long dendrites as well. Neurons are not the most abundant cells in the nervous system, but they're the functional cells. Neurons come in three types. Multipolar, these are the most common types in your brain and elsewhere in your body. Bipolar and unipolar neurons, these are two different types of sensory neurons. I think bipolar are in your olfactory, if I remember correctly, and unipolar are some sensory neurons. Okay, this isn't a big deal, but you should know multipolar are the most common. Most common cells in the nervous tissue are glial cells or neuroglial cells. These are supporting cells. Okay, they do jobs neurons don't do. They help neurons function. They're cells that are, they're accessory cells in there. So like they might hold neurons in position. They form the blood-brain barrier. They act as scavengers. They produce myelin, which your book talks a lot about. Myelin is really important. But these are the different types of nerve tissue are differentiated on the type of glial cells that are present. Okay. Most common glial cells are astrocytes. These are small branched cells. They form the blood-brain barrier. They hold neurons in position. They are important in the development of the nervous system. They guide the migration of young neurons. They make sure the environment for the neuron is correct. They suck up potassium ions. They recycle neurotransmitters. They're involved in the regeneration of damaged neurons, all sorts of stuff like that. You probably hear about astrocytes in cancers, brain cancers. The most common brain cancers are astrogliomas. And so they can occasionally be involved in cancers. Ependymal cells produce cerebral spinal fluid. They line the hollow spaces, what are called the ventricles of the brain, and the central canal of the spinal cord. They have cilia that move cerebral spinal fluid. They look a lot like epithelial cells, but they're not typical epithelial cells. So they're important in the central nervous system. Microglia are the smallest of the glial cells. They're phagocytic. I'm not going to say much more about them. We get to the myelin-producing cells. Okay, in both the central nervous system and in the peripheral nervous system, we have myelin. It's a fatty protein, a lipoprotein that's used to insulate neurons. And here you see this node of Ranvier here. This is where the junction is between two myelin-producing cells. And oligodendrocytes form these sheaths around the axons and dendrites. And then there are these little gaps called the node of Ranvier, and it causes the impulse from a nerve cell to jump from one gap to the next gap, and it travels faster. This is called saltatory conduction. And so myelin is really important. Glial cells that produce myelin are crucial to rapid transmission of nerve impulses. There's lots of diseases that are really problems with myelin. Multiple sclerosis, myelin breaks down. ALS, remember the ice bucket challenge from a few years ago? That was a problem with myelin. That's a disease where the myelin breaks down over time. And so if the myelin doesn't work, people can become paralyzed. They can lose sensory function. Anytime you have rapid transmission, myelin is involved. And this happens, oligodendrocytes do this in the brain, and Schwann cells do this in peripheral nerves. And so Schwann cells, there's some Schwann cells there that are doing the same thing. And you can see the junction between different Schwann cells. That's the node of Ranvier. So really important. Myelin, we won't talk much about different nerve tissues, but we'll talk about white matter, which is myelinated, and gray matter, which is unmyelinated. Most of your peripheral nerves are myelinated. You have myelin tracts in your brain, but then you have lots of gray matter in your brain as well, and that's unmyelinated. Okay. And so here's kind of the thing. Myelin protects the axon, insulates it electronically, increases the speed of nerve transmission. And there's that lower right picture that just shows that really dark stuff labeled MS. That's myelin wrapped around a neuron. So So it's an important function. Okay. So the nervous system controls other organs. It has sensory, motor response, and integration. We have these divisions of the nervous system, and then neurons and glial cells. This is a big important section. Half the chapter really is that. That's half the chapter. Okay. All right. So, anybody have any question? Okay.
I have a question.
Sure, please.
I was trying to take the quiz, the muscle quiz, the lab quiz, and the last question, I saw there was a movie called Muscles, but I wasn't able to watch it because I didn't know where to see the movie.
It's in the box that was last week's box, and so there's a movie called Muscles in there. You have to enter your library card number, and so in that yellow box, if you go down, there's a place for you to enter the number. Okay, and it's just like other movies that we've looked at.
Okay.
Okay.
All right. Thank you.
Ask me tomorrow, please. I can help you with that tomorrow.
Where do we
Okay.
submit? Where do we submit that activity?
The I did not open the box. I'm sorry. I will get that.
It wasn't opened. Yeah. Okay.
Okay.
All right.
All right. So then the other really big deal for us is how neurons function. Okay. And so this is action potentials. And we've talked about action potentials with muscle cells. It's a change in the charge at the membrane. Okay. And neurons have the same type of action potential that muscle cells do. These changes in charge occur when ions move across the cell membrane. The cell, just like muscle cells, maintains a resting potential where the cell normally has a negative charge on the inside and it's positive on the outside. And then there's a gradient, chemical gradient as well, with more sodium ions on the outside and more potassium ions on the inside. And so the action potential occurs when the cell becomes less negative, so it depolarizes. And when it reaches a threshold, which you have here, which is, I think, about minus 60 millivolts, okay, that causes voltage-gated channels to open. Okay, when these open, more sodium ions can enter the cell, and so the cell becomes less and less negative. Okay, and when this happens at one spot in the cell, it causes nearby ion channels to open, and so this depolarization will sweep across the surface of the cell, and the cell will do that action potential, which you can see here is in the graph where it says membrane potential. You see action potential, threshold, action potential. This action potential is very quick. This happens in milliseconds. Okay, and so these sodium channels are open for a very, very small time. And then they close and potassium channels open. Okay, and potassium, which is in excess inside the cell, will diffuse out. Okay, and so this repolarizes the cell. Okay, and so cells will go from being negative to briefly positive back to negative, and that's the action potential. And this spreads from one part of the cell to another, and it's very, very fast. For one cell, even a large neuron, this will cover that cell completely in milliseconds. And so that's the key thing, is that this action potential will start at the dendrite and pass all the way down through the cell body and down the axon to the ends of the cell, so that this charge will go from one end of the cell to the other extremely quickly. Okay, where it winds up is a synapse. And so these synapses are at the receiving neuron. There'll be this junction where one cell ends and the other cell begins. And neurons signal other neurons across this little gap, the synapse. And they can do this electrically, some of them, but most of them will do this chemically. They will release a neurotransmitter. So these little chemicals that are called neurotransmitter molecules will diffuse across the gap and bind to receptors on the postsynaptic cell. Okay, so the postsynaptic cell will have these receptors, and whatever the receptor is linked to, that will be stimulated. And so in a chemical synapse, the presynaptic cell has an action potential that causes a neurotransmitter release. This neurotransmitter release is usually due to calcium channels opening at the presynaptic terminal. Okay, then the neurotransmitter binds to the receptor, and whatever the receptor is linked to will happen. Okay, and so this is just kind of going through it step by step. So you have the 1, 2, 3, 4, action potential, calcium release, neurotransmitter release, and then neurotransmitter receptor binding. The receptor is usually a ligand-gated ion channel that when the receptor binds to it, the ion channels open, and that can cause a change in potential. Okay, these changes in potential, if it's a sodium channel, usually these things are going to be excitatory. If it's a potassium channel, the cell will become more negative, and so it'll be inhibitory. And so that's, there's the excitatory postsynaptic potentials and the inhibitory postsynaptic potentials. And so these are important kind of distinctions to make. Neurotransmitters like acetylcholine here are excitatory typically. And if you trigger enough of these ion channels to open, that can cause an action potential in the downstream cell. In cases like myasthenia gravis, where they have fewer receptors, the release of neurotransmitter has a harder time getting the action potential going because there's not enough ion channels. Okay, one of the great things about the neurotransmitter system is that it can potentially be modified, and so we can affect how long neurotransmitters remain in the synapse. We can break them down faster or cause them to remain longer. We can sometimes add chemicals that will reduce the removal of the neurotransmitter so that they can act longer, or we can add chemicals that act like the neurotransmitter. And so we can do things like that. So in the case of lots of drugs, we add a chemical that can cause the ion channel to stay open or to open longer. Okay, so let me see what I can get down here. So in summary, we have action potentials that are caused by the opening and closing of ion channels. Sodium channels open, those are excitatory. Potassium channels are inhibitory. The junctions between neurons are usually chemical synapses. Okay, in that case, the presynaptic cell releases a neurotransmitter, a signal molecule. The signal molecule binds to receptors that can then be excitatory or inhibitory. Okay, medicines and recreational drugs often affect neurotransmitters. And so there's lots of examples of those. Okay, so there's really lots of stuff here. It's confusing, but in its basic form, it's how neurons work, what is the action potential, how neurons signal other neurons. That's the key thing here. That's half the chapter, but it's the hard half. And so, when you're looking at that, kind of get it down to the basics function and learn the basic function. And it's important to recognize now, because you have excitatory and inhibitory neurotransmitters, this is how neurons can make decisions. Neurons themselves in these individual cells make decisions on what to do based on how many excitatory neurotransmitters are there versus how many inhibitory. And so if you have different sensory inputs coming into the same cell in the brain, that's whether that cell is going to become active or remain inactive depends on what types of neurotransmitters are present and how abundant they are. And your book talks about summation, and that's just kind of weighing, how many are these or how many are of this type. And if there are more of this type, that's what action happens. If it's more of this type, it'll be the other action. Does that make sense? Maybe? Do you have a question, Heather? Lauren?
I don't really have a question other than I think I need to get some more studying, have somebody help me, find a tutor maybe.
Okay.
I didn't get the muscle action potential either.
In that chapter, they go over it really fast because they know, okay, in the next chapter, we're going to go over it again. So they're just kind of introducing the idea. Here we're getting it in more detail. And the key thing is that these ion channels open and close very quickly. And at the synapse, the ion channels, what opens and closes them is the binding of the neurotransmitter. And they call those ligand-gated channels. Okay, but once you get away from the synapse, the other channels are called voltage sensitive. And so they open when there's a change in charge around them. And so once the change in charge gets big enough, it causes nearby ion channels to open. And then it's like dominoes. Once this one opens, then the next one opens, and then the next one opens, and the next one. It's like a domino effect. And so they often portray it that way. Have you looked at the animations? Okay. I would play those a bunch of times. That helps me. It goes fast. Play those a bunch of times and just watch that repeatedly. That's helpful. And there's lots of animations. I pick out the ones I like, but there's others. And sometimes they get too much detail, but the basic process is, first sodium comes in, okay, and you get the depolarization, and then those channels close, and then the potassium channels open, and potassium goes out, and the cell becomes negative again. Okay. Did that help, Lauren? Maybe?
So that brief moment where it's positive in between, that's what draws the charge through to the next one?
Well, so you have to, this whole thing, your neuron is a big, long cell. You have neurons that go from your hip all the way to your toes. Okay. So when it starts to depolarize in one spot by your hip, the next spot will sense that, okay, right next door, the charge is changing. That means I should open and allow sodium to enter here. And so then that charge moves just a little bit, and then the next one gets it. And so that's when I say it's like dominoes falling, it's that once the charge changes here, that causes the ion channels to open next door. That will cause charge to change there. Then the ion channels next door to that will open, and then next door to that, and next door to that. And so it moves, and that movement is fast. It can go from your hip to your toes in a few hundredths of a second. And it has to be that fast, otherwise we'd fall over every time we walked. So that's that really quick. Those are all myelinated channels, so it's happening as fast as we can possibly make it happen. Now, in our brain, we have unmyelinated neurons, and those are slow. Those are very slow. That's why thinking is slower than a muscle reaction type stuff. But watch the animations, play it a couple times. They're pretty short. It's a sequence of events. These ion channels, they just change shape just a little bit. They're opening and closing. They show it as like a little block coming in. They go through a cycle where they open and then they close. The whole thing is open like a millisecond. And we're not talking about a huge movement of ions, just enough to cause a little change in channels. These are millivolts. So it's 1/1000th of a volt, something like that. Okay, it's hard. Okay, it's not the whole part of the chapter though. There's a big part of the chapter that's just sensory response integration. Here are the divisions. And then these are the cell types. Here's how neurons function. So there, those things there. Okay. All right. We'll see how it goes. Okay. All right. So I'll see you Friday. I will be there in and out because I'm kind of in another meeting at the same time. So, all right. Okay. That'll have to be it. All right. I'll post this. Maybe I'll see if I can find another good animation that goes through the whole thing once more, and I'll try and post that too. So, okay. Thank you. See you later.